Okay, so today we have a second part of the second lecture. Before we start, I guess I should perform my promise and show you one more interesting illusion called Ames Room. I have added that somewhere here, I guess. Good. So once again, that's in the context that our brain is processing not like some raw image, but of course adds many things from the context. And since we all are used to straight angles, to some sort of perspectives, so on and so forth, sometimes that may play a little bit of a joke, and we start seeing some things not the way they are in reality. So here you see a video. So here you see two people, and how do you think? Who of them is, let's say, bigger or higher or taller? I don't know. On the left or on the right? Seem equal. Okay. Maybe that is. But let me show you the video itself and look how the how our experience is. So here they stand, and now they start swapping places. And swapping places once again. So maybe you can estimate their like relative height when they were hugging, but just looking at this video, it's really hard to understand who's taller. So that everything boils down to the perspective and to how we experience these sort of things on the background. So actually, they are not straight angles; they are quite different, and the point of view of the camera, of course, is not random, but carefully chosen. But anyway, that's what we see. In olden days, all these sort of perspectives, they were often used to make some sort of effects for movies, some special effects. Well, but that was before the era when we can make anything on a green screen, so I'm not sure if anyone is now bothering with all that stuff, but that certainly worked to a certain extent. Good. Now let's continue where we have ended our journey. So what did we consider yesterday? We started with the photon of light that came to an eye, went through all that, let's say, layers like vitreous body and lens and cornea, so on and so forth, was able to get to the photodetectors, that's our rods and cones, then was detected by our photodetectors. So we have in pretty detailed way considered the biochemical process that leads from getting that photon and transforming that into some sort of cascade of biochemical processes, some signaling cascade that ultimately leads to changes in the, let's say, uh, in the glutamate, in the levels of the glutamate neurotransmitter. And that's, of course, is detected by different neurons, and this information is transferred to the brain. And now we are interested in considering this path to the brain, and basically some, let's say, processing in the retina. It turns out we have certain processes that are performed in the retina, and the information that gets to the brain is a little bit more complicated than just a detection of a light spot. So we will get through that. I will show you maybe in more details what happens in the retina, what is the processing there, because we know a little bit more about the processes in the retina, and then we will consider some general pathways in the brain, how information is spread throughout the brain, and what it tells us. Of course, it's a really complicated task to trace all the things in the brain, and even if you track them, we still are not on the level to completely understand what is happening there in the brain, and how all that information is transformed in some sort of our representation of the surrounding environment. Maybe at some point we will be able to do that, and that will be pretty interesting, because at that point we will be able to copy that into a machine and basically create some true artificial intelligence. And that will be pretty interesting, I guess. Good. So here we are. Here is basically a simplified view of what is happening in the retina. So we are not in the brain, we are just in the retina, and all that layers of the cells, they are performing certain processing. So what kind of cells are we interested in? So here you see our photodetectors. It may be some cones or rods. Here you see horizontal cells, bipolar cells, amacrine cells, and everything gets to the ganglion cells that project their axons to forebrain. So all these things, they all are performing certain processing, and then the signal in forms of, I know, some action potentials gets to the brain and signalizes to the brain that we have seen something. Okay, so what are the points to notice? So with just one exception, the only light-sensitive cells in the retina are rod and cone photoreceptors. So they are the only ones, with just one exception. That's so-called intrinsically photoreceptive retinal ganglion cells, or IPRGCs. You may check that if you wish in the literature, what are they doing, but at the moment, rods and cones are more than enough for us. The ganglion cells are the only source of output from the retina. So these cells are the only ones that are projecting something to the brain and are basically sending some information to the brain. All the rest, they live in the retina, and whatever they do here in place. And ganglion cells are the only retinal neurons that fire action potentials. All other retinal cells, they may depolarize, they may hyperpolarize, they may change some rate of neurotransmitter release. That's okay, but they do not fire any action potentials. So that's one more point to notice. And now we will consider in some more details how this structure works and where it leads us to. I guess maybe Maybe I will show you first this thing, and then we will get back to that sort of simplified view. So here you see something called receptive field. These receptive fields, they can be basically divided into two large categories. One is called on, the other is called off. That depends on the bipolar cell. So that's the cell that will be sending the signal to the brain. Depending on the cell, how it reacts to the signal, the whole field will be called on or off. They are pretty similar, so most cases I will consider just the on version, but you should know there are two. And here you see that our photoreceptors, it's not like one photoreceptor is connected to one bipolar cell and sending some signal to the brain. Not at all. They are collected in this sort of pretty interesting structure. Some of the photoreceptors that come from the receptive field center, they are connected to this bipolar cell. Some of the photoreceptors that are surrounding this part, they are connected to the horizontal cell that is connected to the bipolar cell. And here you see these simplified view of these sort of connections. Now I guess I can return a little bit back, or maybe in here. Yes, maybe I can return here and show you these once again, this simplified version of whatever is happening there. Okay. So here you see once again these sort of structures. So these are circuitry for rods and for cones. Here you see once again these bipolar cells, and different arrows, they indicate some action. So red arrows, they indicate so-called sign-preserving connections. So the higher the signal, the higher is the response of the cell. And gray arrows, they show sign-inverting connections through GABAergic or glycinergic or glutamatergic synapses. So here you see that these arrows, they are inverting the signal, so the higher is the signal, the lower is the response. It looks a little bit complicated, but I will show you some more simplified slides and try to explain you what is happening here. Rod signals feed into the cone circuitry, although the A2 amacrine cell, which serves to split on and off pathways. So here you see A2 amacrine cell, and here you see how that how that proceeds. Okay, so what is this sort of receptive field doing? So here we see cases for on and for off receptive fields. And here you see what is happening depending on the brightness that is perceived with the center of the receptive field or with its edge. So here we have this sort of edge, we have a center, and we may see what is happening here. If there is no light, you see there are some generated action potentials that go to the brain, but they are pretty scarce. When we have light shining in the center and no light surrounding that center, we have a large burst in the firing of action potentials. When everything is lit, we see once again some action potentials are fired, but not many. And the fourth case, when we don't have any light in the center but the surrounding is lit, you see that during the stimulus we have no action potentials fired, so nothing is fired. That's the case of on. Off is just inverted, so you see the inversion here. No stimulus here, large burst in action potentials here, burst in action potentials here, nothing here. So they are just like inverted one to another. So how that's working? I guess it's a pretty it may be a bit complicated to understand what is happening right there. So I tried to create some sort of model that at least explains how these cells decide what should be the signal. These numbers are absolutely made up. So you should not think of them like something measured or something that corresponds to the reality. But this model gives at least some hint of how can we represent in our head whatever is happening right there. So here you see once again some sort of simplified view. I have removed once again some parts of the diagram. And here you see one photoreceptor from the center, so here is the center, one photoreceptor from the periphery, so here you see periphery photoreceptor. Here you see a horizontal cell, and here you see bipolar cell, and this bipolar cell goes to the ganglion cell. What do they do? Well, when there is no light, you know that photoreceptors are generating some glutamate. So you have some signal from photoreceptors when there is no light, and you have no signal when there is light. So if I don't have any light on the central photoreceptor, it's generating a lot of glutamate. Let's say some arbitrary number 4. When we have some light, it generates a little bit of glutamate. Let's say some number 2. And these are the cases for the periphery. So all four possible cases, when you have no light in the center, when you have light in center and dark surroundings, so on and so forth. So all these 4 cases, these are basically these. Oh, what have I done? These are basically these 4 cases, and here we have them. So everything is dark, and everything is lit, and so on. So here they are. Now, what does the horizontal cell? So here we have this horizontal cell. What does it do? Well, suppose it calculates something like this. So whatever it gets from central field minus something it gets from the periphery. And here you see the numbers that we get. And at the end of the day, the bipolar cell that decides whether we are on or off, in this case, inverts the signal. So I have translated that into something like 3 minus whatever it gets from the horizontal cell. So here it is. And now you may check if we have nothing here, so no signal here. We should have some pretty small signal getting to the brain. So nothing here, you see, and we have some pretty small signal getting to the brain. If center is lit and surrounding is dark, what is happening? So center is lit 1, surrounding is dark 0. So when we get to the end of this line, we get 3, so the highest possible signal. And you see that we have really some burst of action potentials here. And so on. You can check these numbers. They are more or less giving some approximate idea what is the output of these sort of things. Now, why is this interesting? The thing is that it turns out that we are not seeing some sort of level of light, so how lit is one point in our retina, but we are seeing some gradients, so some differences between the central part of the receptive field and the surrounding of the receptive field. So we are basically seeing some gradients. And that's pretty interesting. First of all, we are really good with spotting some changes in gradient whenever we are looking at something. I'm not sure if you have ever seen that, but if you ever do, I don't know, some lab works in optics, you will probably meet some devices that are based on the fact that you should make two things equal in brightness. And we are really good with this task. But we are really bad by understanding when something is absolutely bright or absolutely dark. And I want to show you one more example where can we see that. So let me turn to this slide. Look at this figure on the left. How do you think the upper part of this figure, well, it's a bad choice of color, so the upper part of this figure and the lower part of this figure, do they have, is the upper part darker than the lower part? Do you see that or not? Okay, yes, but anyway, I'm not sure how you, but I clearly see some difference in these colors. But if I do something like let me return to this slide. Something like this. So here is our image. Can I do it a bit, I don't know, maybe a bit bigger? No. Maybe this way. Looks good enough. If I take something and cover up the central part, they immediately become undistinguishable. When I get it away, it looks like much brighter. So once again, one of the reasons for this thing is that we do not operate with the absolute values of the brightness, but rather with some sort of gradients. So gradients are calculated in our eyes, so immediately. And that's pretty interesting thing. Let me find where is here is the button for full screen. And that's pretty interesting part, because from these sort of gradients our brain is constructing some subsequent, let's say, features. So like in machine learning, so you have some simple features that are later transformed into some higher order features, and you see that these sort of receptive fields and gradients, they are a method of getting some very simple features that are later integrated in some a little bit more complicated features, and so on and so on. Here is an example of some more complicated feature. That's you see the recording of the microelectrode in the striate cortex. So here you see that, and this sort of recording shows us that there is some neuron that reacts to some lines that are rotated in appropriate way. So here you see this line is differently rotated in the field of view, and here you see the recordings. So cell may completely ignore it, or it may fire some sort of burst of action potentials when the angle is correct. So here you see all the different angles, and here you see how the cell reacts. How do we get this sort of effect? Well, basically by overlapping different receptive fields. So here we have neurons in different cortical layers. I guess I will show you that layers in a couple of slides. when we get a little bit closer to the anatomy of all these things. But here you see that different neurons that get information from the receptive fields, they send signals to another neuron. So here is another neuron, and it can integrate all these signals and construct some sort of receptive field that looks like this. And here you see that this neuron in the primary visual cortex, it responds selectively to line segments that fit the orientation of its receptive field. And the selectivity is the first step in the brain's analysis of the object's form. So here you see once again these sort of images. You see this segment is rotated different way, and depending how it is rotated, you either get a lot of action potentials or you can get none of the action potentials. Here's one more example of constructing receptive field and some sort of responses that we may have depending on the object that gets into that receptive field. So here you see a receptive field that is constructed from three different parts, and let me call them, let's say, these like plus, these minus, and these minus. Whenever something gets into the plus, it has some urge to generate action potentials. Whenever something gets into the minus, it inhibits that sort of generation, and we have no action potentials. Where does it lead us to? If we have stimulus that fully falls in the plus area, we have a lot of action potentials. If we have some large object that falls into both plus and minus areas, we may get no action potentials. Why not? The object may be curved somehow, so it may get into the plus part, but only a little bit into minus parts. Then we will have some action potentials depending on how it gets into there. Or we may have a little bit differently organized receptive field like this one, when we have one plus and one minus, and this thing may be used to detect some sort of corners of the objects. So when a corner gets into the plus area, action potentials are generated. So these sort of neurons, they can be found somewhere in the brain. What's interesting here, this has some sort of machine learning analogy. I'm not sure, did anyone of you interested, maybe did you make any programs that involve artificial neural networks? So especially convolutional neural networks. Does anyone have some experience? Yes, you have. Good. So you should definitely know how the convolution operation looks like. Good. Well, we are not going now into any deep details. So the only interesting part is the convolution operation that basically gives the name to the convolutional neural network. So that was some sort of breakthrough when people were able to come up with this sort of operation. In case you have no experience, so here is an example how that's performed in the convolutional neural networks. So you have a thing called a kernel, and here is your input image or input layer, so depending where this convolution operation is in the neural network. And you might think of it like you are overlapping this kernel with some part of the image. You are multiplying numbers here with numbers here, then you add everything together, and the result of whatever you have obtained, you place into the output image. And so you do for every possible position of these kernels. So you just go along that image and you calculate something and you put some numbers in here. And this sort of operation, if you carefully choose the kernel, that will look pretty similar to whatever we have with our receptive fields. So when we have some signal in the center, the neurons tend to generate some action potentials. And whenever we have some signal on the periphery, neurons tend to inhibit that sort of generation. So that system I have shown you, these sort of receptive field, you can think of it like you have taken a kernel that has, I don't know, some positive number in the center, let's say 5, and has some negative numbers on the periphery like -1, -1, -1, and so on. And this will be basically pretty similar to what is calculated in our retina, if we can call it that way. So these operation in the artificial neural networks, in some sense, mimics the these sort of these sort of receptive fields that we have in our retina. So that's pretty pretty close, let's say, in some sort of philosophical from the philosophical point of view. Good. Okay, suppose we have processed that thing, so we have performed all that operations in our retina, and now we are sending signal to the brain. Where does it go? And it turns out there are many different pathways depending on what we want to do. So here you see part of the pathway that leads to some sort of conscious visual processing. I will show you that later, maybe in a little bit of more with a little bit more details. So here it is. Here you see some pathway for the pupillary reflex and accommodation. So you know that our pupils, they change their size depending on how lit is the room where we are and so on, so depending on that lighting conditions. And this is some sort of unconscious event, so you cannot control them consciously. Well, at least unless you shine with some sort of flashlight in the eye. Otherwise, I guess there's no way. And here you see that pathway, so the signals go to the protectum, then to the accessory oculomotor nucleus, then to the ciliary ganglion, and then they get to the pupil and perform contraction, for example. Here you see example of a horizontal eye movement. Vertical eye movement has certain subtleties, but so let's check this one. So here you see a little bit more complicated pathway, and here we see structures that are involved in the visual processing, in the conscious visual processing, like LGN, that's lateral geniculate nucleus, or here we have so information goes to the lateral geniculate nucleus, and part of the information can go to the so-called superior colliculus. There are like 4 of these colliculi, two superior and two inferior, but I don't think we need to get into all the details. But that's pretty interesting part, that some information gets into that SC and then is processed and may basically through the abducens nucleus and through the abducens nerve get to the eye and make you move your eye. So in some cases, when there is something wrong with the visual processing, a human may be blind, but eyes may still react by moving due to the path through the SC being intact. Rare cases, but but it may happen. So some parts are broken and no conscious visual process may be performed, but this pathway still survives and still may perform some movement of the eyes. And regarding that sort of movement, here is one more example for you. So here is a picture. Now you look at this picture, and do you notice that you move your eyes when you are looking at this picture? So you are not like looking at the picture as a whole, so you are looking at different details and you are somehow moving your eyes to concentrate on different details. And that is pretty normal situation. We all do that, and moreover we can even trace how our eyes move, and here you see one of those traces. So here you see how we move our eyes. It's definitely seen that we are paying a lot of attention to different faces. So some large part of our brain turns out to be processing that faces, so we need that information, and we see that we pay a lot of attention to different faces. So here you see some track of the eye movement during looking at this picture. Good. So now let's get to the visual pathway that mediates some conscious visual perception. So here we see a side view of the brain with the retinogeniculocortical pathway shown inside blue. This complex name means that we start in the retina, we get to the geniculate nucleus, so LGN, and then we get to the primary visual cortex. And from that cortex, there will be more and more projections and so on. And here is some sort of horizontal slides that shows this pathway. So here you see retina, here you see the chiasm. So here information from the right field of view is transferred to the left, and information from the left field of view is transferred to the right. Here you might see the LGN, so these are lateral geniculate nuclei, so the left one and the right one, and these are optic tracts. So please take a note. In some sources, people incorrectly say that information from the left eye gets to the right, and from the right eye to the left. That's completely incorrect, because the correct statement is that information from the left field of view gets to the right, and information from the right field of view gets to the left. And the thing is that both eyes have left and right fields of view. And here you can clearly see that in the image. So half of the retina is projected in each of the eyes, half of the retina is projected to the left, and half of the retina is projected to the right. Well, in some sense. A pretty interesting thing I just couldn't not to mention when it came this way. So you see that we have these fields intersecting. The area they intersect, that's basically the area or the volume where we have some sort of binocular vision. So we can estimate distances to the objects if they fall in this sort of cone, or I don't know, some volume created by left and right fields of view intersecting. And of course I couldn't leave it behind and not show you how that works in different animals. So here you see, for example, a cat. So here is the area of the binocular vision. So you can estimate that like 140 degrees. So pretty large angle. And of course, there are some areas when one of the eyes can get information from that area, while the other one cannot. For example, vision is blocked by the nose. So we have a nose, animals have a nose, and it blocks some part of the vision. So anyway, you can't get information from everywhere. Here is the this sort of area for the dog. And interesting part is that these animals, they are mostly carnivorous, and they have this binocular vision in front of them, so they can clearly see some other animals that they may want to catch. While there are animals like herbivorous animals that are not trying to attack anyone, but that need to see everything around them and being prepared to flee when some of the carnivorous animals attacks. And here is a great example of a rabbit or a hare, I guess it's a hare, uh, which has so large fields of view that he basically sees all everything around, so all 360 degrees. So here you see there are some blind areas, so you definitely cannot see what is behind you, so we have some blind area there. These animals do have, while hare has his eyes on the sides of his head, and he's basically seeing everything around. Moreover, these sort of binocular areas, so he's seeing so clearly everything around that he has even some binocular area behind him. Well, pretty small angle, but still something can fall into that. So he can see things binocularly in front and behind him. So that's a pretty interesting thing to notice. So that may happen in nature. Okay. here we go further. Here you see this sort of processing. So we have retina, we have some projections to the LGM, to the geniculate nucleus, and we have then projections to the striate cortex, to the layer 4C. We will get to these sort of numeration a little bit later. So the thing is that neighboring locations on the retina, they project to neighboring locations in the LGM. This retinotopic representation is preserved in the LGN projection to the V1, that's a layer of the visual cortex. The lower portion of V1 represents the top half of visual space. So here you see these sort of representations, so the image that eyes see, and here is how they are represented in the visual cortex. So you can see it's like turned upside down and looks maybe a little bit strange. So the map is distorted with more tissue devoted to analysis of the central visual field. So we have already considered that we have this sort of fovea, and we have some field where we see the most clearly, we see some colors, and that's basically the largest part of the brain is devoted to the processing what is basically there. We can have some similar maps in superior colliculi, LGN, and some other cortical areas. Here is one more mapping how the information gets from eyes to the visual cortex, and what happens to the visual field in case anything is broken. So if we have some damage to the nerves somewhere along this path, you see that different parts of the visual field may be broken. So something is wrong. So here you see the image that gets into the left eye, into the right eye, and basically what happens if anything is broken. So that's if optic nerve gets damaged, that's pretty obvious. You just don't get any signal from the eye. If you have the chiasm broken, you see that part of the field that should be transferred from the left eye just does not reach the processing areas in the right, and parts from the right eye don't reach that areas on the left, so some parts of the image are missing. And whenever we have this optic tract damaged, you see once again some parts are missing, but you see the picture is a little bit different. Yeah, so here you have one part of the field, so either that's the right part, so you should have the left part of the visual field, and if here is damaged, you have this part just broken, it just goes away. And here you see that in the image. And of course, after the LGN, there's a little bit more little bit of a processing, and then different neurons are projecting into the visual cortex, and if you break anything there, some smaller parts of the visual field, they will be not visible. The center in some cases seems to be okay because the representation of the foveal stimuli is projected to both hemispheres with various degrees of the overlap. So in these cases, like when you damage here 5 and 6, the brain is still able to recover at least this central part. So as you see, we have a lot of processing there, and the brain is trying to cover up for whenever anything goes wrong. Now here we see the LGN, that's of the macaque monkey, and here you see some sort of layers in that LGN. So here is the slice, so that's the brain, the whole brain. Here you see the slice. Here in this slice you can see the LGN, and if this tissue is stained to show cell bodies, you may notice the 6 principal layers and the larger size of the cells in the 2 ventral layers, like layer 1 and 2. So here are 6 layers, and here they are numbered like 1 to 6, and here they all are. I guess I have, yes, I have one more picture here that shows you how this architecture corresponds to different visual fields. So here they are once again, 1, 2, 3, 4, 5, 6, so 6 layers in the LGN. And here you see 2 eyes. These are like projections from the retina. So here you have right temporal retina and right nasal retina. So nasal means closer to your nose. Here the same for the left eye, so nasal and temporal. Here they go through the chiasm. So here they are resorted a little bit. And here's how they are distributed through the different LGNs, through the right and through the left. So here's the let's say architecture, how the information is transferred. Some more details are in this table, but let's not get so deep inside. Here's once again a view of the LGN and the information that we get. So here is LGN that gets information from the optic tract. Here are the monocular and the binocular zones. You see that information comes from both eyes, but that's information corresponding to, in this case, I guess, left field of view. So here it is. Here you see how this information gets into the LGN, and you might notice that different layers of the LGN get information from their respective eyes. So like layers 1, 4, and 6 will get information from the left eye, while layers 2, 3, and 5 will get information from the right eye. So it's not like some sort of mix of both eyes, but here they are still separated on this level. Later, everything will be collected together to build some sort of model of the outside world, but at this stage in the LGN they are still separate. Then they are projected to the primary visual cortex, so here it is, And here you see the structure of that visual cortex. So here is 1V like visual and C beta, 1V C alpha, and so on. 1V alpha, 1V B, 1V A, 1V B, and so on. And here you see that projections from the LGN. How do they get here? So layers 1 and 2 are projected to the to this to CA to the C alpha, and other layers are projected to C beta. Here are differences in neurons that we have in this area. So here's once again different layers, and here you see connections between these neurons. Maybe that's once again too detailed, but this picture probably is worth looking at. I guess yes. What do we have here? Once again, we have information from retina through the LGN to V1, then it gets to V2, MT, and so on. So it gets to different areas of the brain. And here is what's happening once again. So here we have retina. We have different ganglion cells. So some of them are midget ganglion cells, some of them are parietal ganglion cells. So whatever. Their connections are called appropriately magnocellular and parvocellular. So some of them are, so they are connected to different layers in the LGN. Then they are transferred to the primary visual cortex. So here you see that. And what is interesting, the different parts of that visual cortex, so we have still some sort of lower, let's say, not so complicated processing of the images. They exhibit certain preference to different directions of the objects that we may have. So here you see these preferences on the on this layer, and then you have these preferences on the upper layer, like on second, on third, on 4B, and so on and so forth. So here they are, and you see that this information is then transferred to different parts of the brain, to the V2, V2, to the V4, so on and so forth. And depending on where we are processing different aspects of that information. So here you see procession of the direction. So here it is. Here you see the processing of the orientation. You may see processing of colors. So some colors are processed in here. You may see processing of the depth. So depth is processed in V2. So here it is, the sixth stripe in the V2. You may see some processing of the complex forms. So that's more on the level V4. So depending on the level, we have different aspects of the objects processed. Here is one more image. So here is the stimulus. So many different stripes, and when the eye sees this sort of image, it gets once again through that LGN, so on and so forth, it gets to the primary visual cortex, so V1, and here you see this pattern that is some sort of excitation pattern based on the stimulus seen. And here you see once again how that looks like under the microscope when we consider areas V1 and V2. And here you see different eyes, like left and right eye, and so on. So all these stripes, they are seen in the brain as some areas of activity when we see certain stimulus. Here is a bit more detailed view of these sort of different areas, how different brain neurons are basically reacting on the stimulus depending on its orientation. So here orientations, they are defined with some sort of color. So here you see how the object should be oriented and appropriate color. And here you see that in V1 we have areas that are more like reacting to objects oriented that way. So here are some sort of micrographs, and you can see different views of what is happening there. These one show the orientation. This one, I guess, shows the cytochrome oxidase. That's from the electron transport chain, the so-called complex 4. I hope you remember what was that. So we have considered electron transport chain. Good. And well, basically that's some sort of architecture of that visual areas. Here is a more complex scheme. So whenever information was transferred to the V1, where does it get from that V1 and all the different pathways for our, let's say, processing of the information. I will not get through all these areas. I just want you to know that we have certain ideas how the neurons are wired and how the information gets from one part to another. Of course, there are many details to be filled in, and we still don't know all the details how this processing is performed and how do we get what we know about the world and some sort of consciousness. But anyway, some pathways are known, and we know that some damage to certain pathways may lead to some interesting effects, let's say. I have an example for you. Yeah, I have at the end of the lecture, I have an example for you, but we will get to that. I guess you will like it. Good. So here is one more example. So here you see the inferior temporal cortex. So on the previous slide, here it is. So this IT, that's inferior temporal. So that's inferior temporal cortex. And in this part of the brain, if we use some sort of recording from that part, recording of the action potential, we may see that it somehow reacts to the phases. So at this stage in the brain, we see that our very simple features. So we started with some sort of gradients, and then we saw that different parts of the brain, they have certain preferences to the orientations or something like that. But when we get to the inferior temporal complex, we see there is certain preference to the phases. And here is result of the recording from some sort of, I guess, maybe some macaque, monkey, or something like that. And it was shown some images of other monkeys or human or something like that. And here you can see, so here is the face of some monkey, and here you see some burst in the activity. So you have many action potentials that can be recorded from this part of the brain. Now you see this face was defaced so it was changed so that you don't recognize it as the face, and you see that activity has dropped down significantly. So that's a significant drop down from whatever we had here. Here's once again face of a monkey, once again a burst in activity. Now some features of the face were masked. So suppose we remove some part of that image and we mask, I don't know, either eyes or some lower part of the image, so whatever, some mouth of the monkey. And here you see there is certain activity, and basically if we look, we certainly recognize these are faces. So when I look on this thing, I don't recognize it as the face, so it requires certain amount of work, some mental work to understand what that. These are definitely recognized, and you see in case of monkeys we still have some activity, but this activity is somewhat lower than in the case of let's say fully seeing some face. We can remove some color. So here's once again a face of a monkey but without any color. We still have some burst in activity, so which tells us that even without color, there's enough information from the for other areas of the brain to transfer here some information that will be a face. So these neurons are still recognizing that as a face. Here is the human's face. Well, not very large activity, but still seen that it can be somehow recognized as a face by the monkey. And here is a completely different object, like an arm. So you have a hand here. So just with looking at the hand, you see almost no activity because it's not a face. So we know that in the inferior temporal cortex has some neurons and some circuitry for processing faces and for recognizing whether the object is the face or that's something pretty different from the face. And I guess let me just check for a moment, where are we? So I can show you. We're almost there. Okay. Yes, I will show you few nice images, and then we will return to this sort of to this inferior temporal cortex. So yes. So don't forget this part. In a moment I will show you something interesting regarding to that. Just few more images for us to show that when some parts of the image are missing, our brain is still able to process everything and give us some general picture that involves even missing parts. So probably on the left you are able to see a white triangle. So I guess you should be able to see that. And of course we all understand that there is actually no white triangle, so these are just some breaks in other figures. Here we clearly see some sort of red square, despite the fact there is no red square. Even the area in between these circles may look for you a little bit reddish, but there is definitely no red square. Only some parts of these circles were colored in red. So that gets along the lines. So if we have some parts of the image removed, our neurons may still react on some higher levels of processing, may still react to that as the whole image. So that's the case with monkeys when we have removed some features from the images. Let's say we hit their eyes, for example. One more interesting thing for you. So here is an interesting effect. I'm still not sure how that is explained, but that's an image for you. So let's check once again whether you're able to see that or not. Do you see any pattern here? So anything, or just different I don't know, some shatterings of something? No. So you can see anything here, but if I hide some part of these, you will be definitely able to see something. Now can you tell me, do you see anything here? So any pattern? B's, yes, absolutely correct. There's a lot of letters B in here, and that's basically the same image. So the previous image and this image, they are the same. Maybe I had some troubles by positioning them, but anyway, if you take these images and compare them somehow, you will see that these are the same shatterings, but just some parts are hidden behind this sort of dark thing. And now you can see different letters B. So hiding even some part can be helpful in some cases. Which is pretty interesting. Okay, what else do I want you to show? So here we are getting to another interesting part. So here are different parts of the brain that, when damaged, lead to certain maybe unexpected consequences. So in case of damage of this part of the brain, and you can check this map, so you can check this map and find that areas, well, to more or less understand what's happening in there, leads to so-called apperceptive agnosia. So that is the loss of the ability to copy or match visual stimuli. And here you see the visual stimuli that were shown to the patient, and he was asked to copy them. And here you see the patient's drawings. So they totally do not look like whatever he was shown. What's interesting, if you ask him what is he seeing, he will give you some clear answer. So I'm seeing a circle, I'm seeing a square, a diamond, I'm seeing number 3, number 4, but he's unable to draw them. So that's what happens if this area is damaged. In case, so that leads to the fact that these patients cannot see objects' parts as a unified whole. And that's why they are unable to construct sensory representations of the visual stimuli. Maybe another case, if we damage this part of the brain, so in the lower image, that leads to so-called associative agnosia. Here you see once again the patient is shown some models, and pretty complicated. So some pen, some tea bag, some ring, so looks pretty complicated. He is able to draw all that, but he is unable to tell you what kind of object is that. So he can draw you a tea bag, but he cannot call it a tea bag. He does not know what that is. So this sort of damage may lead to the fact that people cannot interpret, understand, or assign meanings to objects. So sensory information representation is created normally, but cannot be associated with some meaning, function, or utility of the object. And that's a pretty interesting case. I want to show you, I want to cite you some part of the book directly about these associated visual agnosia. That's a book of Oliver Sacks. It's called "The Man Who Mistook His Wife for a Hat," and I guess and other clinical tales or something like that. So you can Google that. He does not give any, let's say, information on the diagnosis. So this is just some sort of guess from whatever he is describing, but the case he described perfectly fits into the associative visual agnosia. And I just want you, since we are finishing this lecture, so at the end, I just want to give some sort of feeling how that looks like if you have a patient with certain damage of certain areas in the brain. In this case, the associative visual agnosia. So there is some probable diagnosis, the posterior cortical atrophy, but anyway, so what's the case? So there was a man, so he called him Dr. P. He was a musician of distinction, well known for many years as a singer and then at the local school of music as a teacher. So that's a highly intelligent man, so so you will not expect some extraordinary behavior from him. And whatever happens, this Dr. P, he goes to the Oliver Sacks, and he says that he has some vision problems. And he asks him, so why are you here, so and so on. And he tells to Sachs that I was with different doctors. I told them that I have some problems with vision and so on and so forth. And at the end of the day, they told me you need to consult with the neurologist. So he went to Sachs and he told him, yeah, I need to consult neurologist. I have some vision problems. And he asked him, what kind of vision problems do you have? And he says, well, it looks like I see, but sometimes I cannot recognize, let's say, my students, so I can't recognize them, despite the fact that I should know them all very well, but still I can't. Some other things. And so Sachs tries to test him, doesn't find any problem at the first sight, so everything looks like pretty okay. He doesn't really know what to do, what to tell, because everything seems to be okay. But now this happens. So I will recite this part of the book. So he says, I had taken off his left shoe and scratched the sole of his foot with a key, a frivolous seeming but essential test of a reflex. And then, excusing myself to screw my ophthalmoscope together, left him to put on the shoe himself. So he's now sitting without a shoe. To my surprise, a minute later, he had not done this. Can I help? I asked. Help what? Help whom? Help you put on your shoe. Ah, he said, I had forgotten the shoe. Adding sotto the shoe, he seemed baffled. Your shoe, I repeated. Perhaps you'd put it on. He continued to look downwards, though not at the shoe, with an intense but misplaced concentration. Finally his gaze settled on his foot. That is my shoe, yes? Did I mishear? Did he missee? My eyes, he explained, and put a hand on his foot. This is my shoe, no? No, it's not. That is your foot. There is your shoe. Oh, I thought that was my foot. So here are seeing his foot, here are touching his foot, and he is seeing his shoe. And he does not understand what is his foot, and what is his shoe. So that's looks pretty crazy, I guess. So he clearly sees, but he has some damage to this sort of area. So he has some form of the associative agnosia. So he cannot understand or assign meaning to objects. He cannot assign meaning of the foot to his own foot and meaning of the shoe to his own shoe. That's not all. There are some other interesting parts from that book. So I guess we have only 2 slides left, so let me be a bit frivolous and read it to you, so maybe you can appreciate how strange that looks like and what strange machine is the human's brain. When something is broken, it leads to these sort of consequences. So there is one more part. So that's basically a very long story of the Sachs. I have just copied some parts, I guess. And there is a part when he comes to the house of his patient and starts some sort of testing. So he's showing him some images and asking different questions. So what is this? I asked, drawing out the first one. A cube, of course. Now this, I asked, brandishing another. He asked if he might examine it, which he did swiftly and systematically. A dodecahedron, of course. And don't bother with the others. I'll get the icosahedron too. Then, after some more tests, there is one more interesting twist. So when Sachs went to this patient, he stopped at a florist, and here how he writes. I had stopped at a florist on my way to his apartment and bought myself an extravagant red rose for my buttonhole. Now I removed this and handed it to him. So maybe you remember that old fashioned, like wearing that flowers in the buttonholes. Never have seen that myself, well, except for some movies. So he took the rose, and like a botanist or morphologist given a specimen, not like a person given a flower. About 6 inches in length, he commented. A convoluted red form with a linear green attachment. Yes, I said encouragingly. And what do you think it is, Dr. P? Uh, Not easy to say, he seemed perplexed. It lacks the simple symmetry of the Platonic solids, although it may have a higher symmetry of its own. I think this could be an inflorescence or flower. Could be? I queried. Could be, he confirmed. So he does not recognize the rose, even having that in his hand. Smell it, I suggested. And he again looked somewhat puzzled. So, yeah, why do you ask me to smell some strange thing you have handed? Okay, so he's smelling that. He complied courteously and took it to his nose. Now suddenly he came to life. Beautiful, he exclaimed, an early rose. What a heavenly smell. He started to hum Die Rose, Die Lily. Reality, it seemed, might be conveyed by smell, not sight. So you see some parts of the brain are damaged. He's not able to integrate the visual or tactile information, but the smell, you remember some oldest parts of the brain, so we have considered that that is connected to the smell, and you see that they are like helping him a lot to get the correct information out of his brain, so to understand that this is a rose and nothing else. And just another last excerpt I have here, so once again to make you ponder so how strange is our brain and what strange things it's able to do, basically, especially when something goes wrong. What is it? I asked, holding up a globe. May I examine it? He asked, and taking it from me, he proceeded to examine it as he had examined the geometrical shapes. A continuous surface, he announced at last, enfolded on itself. It appears to have, he hesitated, five outpounchings, if this is the word. Yes, I said cautiously. You have given me a description. Now tell me what it is. A container of some sort? Yes, I said. And what would it contain? It would contain its contents, said Dr. P with a laugh. There are many possibilities. It could be a change purse, for example, for coins of 5 sizes. It could... I interrupted the barmy flow. Does it not look familiar? Do you think it might fit a part of your body? No light of recognition dawned on his face. Later, by accident, he got it on and exclaimed, "My God, it's a glow!" This was reminiscent of the Kurt Goldstein's patient Lanuti, who could only recognize objects by trying to use them in action. So here you see a really strange case of someone with the with some damage of probably this area of the brain. So whatever was the reason, and you see that that leads to the inability to associate different parts and different modes of perception into one coherent object that will be represented in our mind, like so sensory representation is created normally, but it cannot be associated with meaning, function, or utility. So he's not retarded, so he knows pretty complicated words and can describe what is a glow, but he cannot understand that this is a glow and it should fit his hand. So he's creating some hypothesis, so why not use it as a purse or something like that? And so that's the strange case. Okay, I guess with that being said, I have used up all my time.